Hello friends and welcome to CEC live lectures. In the ongoing lecture series on extraction of natural products, we started with enzyme assisted extraction, and I will just give you a quick recap of what we discussed in my previous lecture. We started with the discussion on the components of cell wall. What are the various components that constitute the cell wall which provides the mechanical strength and protection to the plant cells. Then we discuss about the principle of enzyme assisted extraction, that is how enzyme assistance in extraction enhances the overall yield of the extraction process. We also discuss about various enzymes that are involved in this technique, like cellulases, hemicellulases, pectinases and proteases. We also discuss about various examples while discussing the different types of enzymes and how they act upon the cellular components. So friends, in continuation with the previous lecture, today I am going to discuss the other aspects of enzyme assisted extraction, and we start with the steps that are involved in this process. So the first and foremost step when you are going to perform enzyme assisted extraction is the processing of your raw material, and the processing of raw material largely includes the drying as well as powdering of the raw material or the plant material. Obviously, drying has a major impact on the constituents of the plant which is under extraction, as we have learned that tremendous increase in the temperature during drying processes to enhance the drying rate have a negative impact on the chemistry of the chemical constituents. Then comes the powdering or the size reduction step of the plant material. Here also the technique that is being used to powder the drug has a great impact on the overall chemistry of the plant material, because lot of heat is generated whenever you are powdering something, and that heat may lead to the degradation of the chemical constituent. So one has to be very careful in such processing steps in order to preserve the natural chemistry of the plant material. So once we have processed the raw material, next step is the selection of the enzyme. As we discussed yesterday that cell wall has different components ranging from cellulose, hemicellulose, pectins, lignins and proteins. We have to select the enzyme after understanding the chemistry or the composition of the plant matrix, because this will largely impact the overall efficiency of this process. So once the enzyme is selected, we have to understand the temperature and pH conditions that are required for optimum action of that or optimum activity of that enzyme, because enzymes are nothing but protein molecules, and we know that proteins are the structure of the proteins, and in case of enzymes, the activity of enzyme is largely dependent upon the operating temperatures as well as the pH of the media. So after selecting the temperature and pH, we go for the incubation of the enzyme as well as the raw material. Now when we talk about the incubation, the time of incubation is important. As we will discuss ahead under the factors that affect the extraction efficiency of this process, we will see that the incubation time or the extraction time has an impact on the yield of the product that is obtained after extraction. So next step is the incubation. So once the incubation time is over, then we have to deactivate the enzyme, and this deactivation can be done by various methods, by centrifugation or by adjusting the pH or by increasing the temperature. So once the enzyme is deactivated, the extract is separated by filtration or centrifugation, and that extract is then used for further analysis. So this was a bird's eye view or an overview of the steps that are involved in the enzyme assisted extraction. Here you can see in this picture the flow diagram of the overall process or the steps which we have discussed just in the previous slide. Here you can see the different types of raw material, like seeds are there, fruits are there, roots, bark, leaves or flowers are used as raw material depending upon the constituent or the plant we are using. And these different parts of the plant are dried and powdered. Then the powdered material is subjected for the enzyme assisted extraction after the proper concentration of the enzymatic mixture, proper temperature and pH conditions are maintained. So once the extraction is over, we centrifugate the mixture in order to separate the enzymes and in order to separate the mark, and then the extract is filtered. Now once the extract is filtered, this filtrate is then further used, can be further subjected to the drying process, which we call it as concentration, and this is largely done by drying the extract under vacuum, or we can also go for lyophilization depending upon our requirement, and the end product is the desired material or the extract of the plant. Now we move on to the advantages of enzyme assisted extraction. There are numerous advantages, and not only advantages, there are disadvantages or limitations of this technique as well, like any other extraction technique. So as we know that enzymes, they act at a specific temperature and pH conditions, and normally we prefer enzymes which undergo which exhibit their activity under mild conditions, because such mild conditions will not impact the chemistry of the plant material or the constituents that are present in the plant material. So therefore, since we have to select the enzymes which work under mild condition, this is one of the advantages, that this process, the whole process, undergo under mild conditions, mild reaction conditions. So such kind of mild reaction conditions help in preserving the natural chemistry of the constituents that are present in the plant material, and eventually these constituents will be coming in our final extract. Then by using enzyme assisted extraction, we have a possibility of using the entire plant material. When I say entire plant material, it means we can avoid the step of powdering or size reduction of the raw material. No doubt, by powdering a raw material or by size reduction of a raw material, we increase the surface area of that material, which further enhances the overall extraction efficiency. But in case of enzyme assisted extraction technique, we have to incubate that material along with the enzymatic solution or enzymatic mixture. So in such cases, in such scenario, we can always go for using the entire plant material without powdering also. But as I told you that we should go for powdering because it will further enhance the surface area and will have an impact on the extraction efficiency. But we can always go for the entire plant material as well. This is another advantage of enzyme assisted extraction. Now if we go back to the steps that are involved in this process, we will see that this kind of technique or this kind of process requires fewer number of steps. So we can say that the enzyme assisted extraction occurs quickly, as well as there are fewer steps that are involved. So the overall time taken to extract the plant material, to handle the material, to concentrate the material overall reduces, and this makes this extraction technique more efficient. Another advantage is the quality of the extract that we obtained. Initially we discussed that this process undergoes under mild conditions in order to preserve the natural chemistry of the constituents. Another advantage is that since the enzymes have already degraded or hydrolyzed the cell walls, lesser amount of solvent is required in order to extract the constituents, and therefore the end product, that is the extract, will have very low levels of solvent residue, making or improving the overall quality of the end product. Then this technique also exhibits a lower production cost. Since the number of steps involved in this process are low, the overall instrumental setup that is required, that we have discussed in numerous modern techniques as well as the conventional techniques, is not required in enzyme assisted extraction. So by and large, if we see, the entire cost of the process decreases when we are using enzyme assisted extraction in comparison to the modern techniques. We also discussed that enzymes have a high specificity. Specific enzymes, they hydrolyze a specific substrate, and therefore this type of specificity will help in reducing the amount of non-required constituents in the final product. Such kind of specificity in turn leads to extracting a large number of bioactive compounds, the required bioactive compounds that are obtained, or we can say the bioavailability or the availability of such constituents increases when you are using this technique. This is a time efficient process, we have already discussed, and it reduces the solvent consumption. So these are the few advantages of enzyme assisted extraction. Now we move on to the limitations of this technique. In my previous lecture, we discussed about the overall chemical composition of the cell wall, and we found that cell wall is a multi-component structure. So over the period of time, it has been observed that the enzymes that are used to hydrolyze the cell wall, they exhibit an incomplete hydrolysis of the cell wall. Though this hydrolysis definitely impact the overall extraction yield in a positive manner, but this incomplete hydrolysis also act as a limiting step or limiting factor in the extraction yield of the compounds. Then if we use this technique at commercial level, wherein we are using huge and large amount of the raw material for processing, the overall cost of the enzymes that are used, and moreover the cost of the enzymatic mixtures that are being used to process that raw material increases significantly, which sometimes make this process costly. Another limitation is the difficulty of this process for scaling up. We know that the process that is used at laboratory scale has certain set of conditions, but when we try to use that process under large scale conditions at an industrial or commercial scale, definitely we will see specific amount of difficulties, because when we move from laboratory scale to industrial scale, the temperature conditions, the volume of the solvent or the solvent conditions changes altogether. The pH conditions changes altogether. That will hamper, that will affect the enzymatic hydrolytic activity. So it's very difficult to optimize when we move from laboratory scale to commercial scale in case of enzymatic assisted extraction. And when we go for the commercial scale, the extraction, it has been seen that the extraction time exceeds tremendously, increases tremendously. So it leads to longer extraction time. So these were few of the limitations of enzyme assisted extraction. Now we move on to the factors that affect the overall efficiency of this process. Like any other extraction technique, this technique has certain factors that will have a positive as well as a negative impact on the efficiency of this process, and these factors include the type of the enzymes that are being used. Though we are going to discuss each and every factor in detail also, and so the type of enzyme, as we discussed, the cell wall is a multi-component structure. So depending upon the chemistry of the matrix, depending upon the structural composition of the matrix, we have to select the enzyme. Second factor is enzymatic mixtures, and since the plant matrix again are a multi-component structures, different enzymes are mixed together in order to get better results. So what kind of enzyme mixtures are being used, what is the composition of that enzymatic mixture will largely impact the efficiency of this process. Another factor is extraction time and the concentration of enzymatic mixture or enzyme solution which we are using. We have discussed extraction time as a factor in various other extraction techniques, and we saw that increase in time enhances the extraction yield, and but up to a specific level. Beyond that level, if the extraction time is increased, we have seen in many cases there is a decrease in the extraction yield. Similarly, the concentration of the enzymatic solution which we are using will have an impact on the overall yield of this process. Then we have effect of temperature on the extraction process. In the previous techniques, we saw that as we increase the temperature, or as a general principle, if we increase the temperature, the viscosity of the solvent decreases. And as the viscosity decreases, it enhances the penetrability as well as solubility of the solvent, thereby enhancing the extraction efficiency. But again, bottom line is the increase in temperature should be up to a critical level. And last but not least is the pH at which the extraction is being carried out. Enzymes are nothing but these are the protein molecules. So their activity will be largely influenced by the pH of the media or the extraction media which we are using. So these are the few factors that can affect the efficiency of enzyme assisted extraction process. Now we will discuss each and every factor with suitable examples also. Coming on to the type of the enzyme that is being utilized in this process. Now it has been observed that the hydrolytic capacity and substrate specificity will have an impact on the overall efficiency of this process. When we talk about the hydrolytic capacity of an enzyme, it will depend upon the specificity of the substrate or that particular enzyme. Say for example, if we have a plant matrix which is rich in pectin. So as a thumb rule, we should use pectinase as an enzyme in order to hydrolyze that matrix. But if you use cellulase enzyme instead of pectinase, then definitely the results will not be as per our expectation, because cellulase enzyme will not hydrolyze the pectin rich matrix. So this is the impact. The hydrolytic capacity of cellulase will tremendously decrease and overall extraction process will become inefficient. So this is how specificity and hydrolytic capacity are related. So in a study, when a cellulase enzyme was used to extract out flavonoids from ginkgo biloba leaves, it was found that the yield of flavonoids was much higher when cellulase enzyme was used in comparison to when pectinase was used. Similarly, cellulase assisted extraction of ginger oleoresin and 6-gingerol, these are the two major components of ginger, it was very significantly higher in comparison to when a mixture of enzymes which constitute alpha amylase, pectinase or protease was used. And in this study specifically, the group extracted ginger with cellulase enzyme, with the mixture of these three enzymes, amylase, pectinase and protease, as well as they also extracted ginger individually with these three different enzymes, and they found the maximum yield was obtained by using cellulase enzyme. In another study, it was noted that the effect of enzyme specificity and the mixture of enzyme on the proanthocyanidin content that was extracted from grape seeds, and it was found that when pectinase alone was used, the yield of proanthocyanidin was much higher in comparison, or it was almost similar when a mixture of enzymes was used, mixture of enzyme which consist of cellulose, tannase and pectinase. So such kind of examples clearly show that when you are selecting an enzyme, one has to understand the structural integrity or structural chemistry of the plant matrix in order to facilitate the enzymatic action by selecting appropriate enzyme. And this not only helps in increasing the overall extraction efficiency, but it also helps in reducing the overall cost. So I'll take you back to the last example which we discussed, wherein the pectinase extraction done by pectinase shows comparable results when in comparable results to that of an enzymatic mixture. So that has an impact on the cost of the extraction process, because mixtures of enzyme are always costlier than the single enzyme. So this is how you can always select a proper enzyme in order to get the desirable results. Next factor is the enzymatic mixtures. Now, as we discussed that the matrix, plant matrix or cell wall is a multi-component structure, and it has been observed that there are various factors that affect the hydrolytic properties of these mixtures. And first one is the composition of plant matrix or the cell wall. Second is the enzymatic activity. Now, as we discussed that enzymatic assisted extraction occurs at specific temperature and pH values. So therefore, if you are using an enzymatic mixture, we have to consider such conditions under which the enzymes that are present in the mixture exhibit optimum activity. The temperature and pH conditions should not be such that they favor only one or two enzymes that are present in the mixture, because this will have a negative impact on the overall efficiency of that mixture. Then the third factor is the types of their constituent and their respective proportions. When we say type of the constituent, which kind of enzymes that are involved in that mixture, and what is the proportion of their of those enzymes in that mixture. Say for example, if a plant matrix is rich in hemicellulose and we are using a mixture of enzymes that is made up of cellulases and proteases and pectinases. So no hemicellulases are there in that mixture. So that will have a negative impact on the overall efficiency of the process. So if we add hemicellulases in that mixture, this will definitely enhance the overall extraction efficiency. This is one aspect. Second is the proportions. Say hemicellulose rich matrix is there and we are using a mixture of cellulase and hemicellulase enzyme, but the composition or the proportion are 2:1. So the concentration of hemicellulose is half with respect to that of cellulose. Again, the extraction efficiency will be affected. So we have to be very careful while selecting, while designing the enzymatic mixtures. And it has been desired that whatever type of enzymes you are using in a mixture, their activity should be complementary to each other. The component enzymes should have a synergistic activity, then only you will get a desired result throughout the process. So again, there are few examples which I would like to discuss that indicates the importance of the enzyme mixture. A group studied the effect of extraction on the yield of the oil by using different enzymes. In first scenario, they extracted the oil by using thyme. They extracted the thyme oil by using cellulase enzyme. Then in the second scenario, they add hemicellulase to that mixture, and they found when they add hemicellulose, the yield of the oil increased. They use similar mixture of cellulose and hemicellulose to extract oil from another plant, that is rosemary, but that mixture did not give any improvement in the oil yield. And when they further studied what was the reason behind it, they found that the trichomes, glandular trichomes in which oil was stored, in case of thyme, the trichomes were covered with a very thin and porous layer of cuticle. So when the cuticle layer was porous, the pores allowed the enzymatic mixture to enter and hydrolyze the wall, thereby releasing and increasing the oil yield that was extracted. But in case of rosemary, those glandular trichomes were covered by a hard and full layer of cuticle. So no pores were present, the layer was very hard, and therefore that enzymatic solution or the mixture was not able to enter into was not able to hydrolyze the cell walls, thereby no effect on the oil yield was observed. So they also did the analysis of that oil, and they correlate the yield of the oil with cravacol content. and you can clearly see when the oil was the plant was extracted by using hydrodistillation, that is the control sample, and then they then they extract the oil by using cellulose individually, then hemicellulose. You can see there is hardly any difference when they use two different enzymes individually, but there was a significant increase in the overall oil yield as well as the yield of Crevacrol when the mixture of enzymes was used. So when they again analyzed the oil of rosemary, and the yield of the oil was indicated by the content of 1,8-cineol, you can clearly see that there is hardly any difference in the oil yield and the cineol yield when the oil was extracted by simple hydrodistillation as well as by using the combination of the enzyme mixtures. So this is how the understanding of the matrix is very important if you want to make this process successful. Here is another example where different enzymes, a mixture of different enzymes was used in order to extract the phenolic components. So this group, you can clearly see in the graph, they use different enzymes, a mixture of different enzymes like cellulose, pectinase and beta glucosidases, and they extract the plant material by different techniques, that is conventional heating method, which is represented by gray colored line, then ultrasound assisted extraction, that is represented by the orange line, and enzyme assisted extraction, that is represented by blue line. You can clearly see that the conventional extraction technique had the content of the phenolic compounds extracted was very significantly lower in comparison to that of enzyme assisted extraction. So friends, in the next lecture I am going to discuss about the other factors that can impact the overall extraction efficiency of enzyme assisted extraction. Thank you.